The broadcast is now starting. All attendees are in listen-only mode.
Good morning and Happy New Year. I hope everybody had a blessed holiday season and are off to a great start for 2016. Thank you for joining our Metabolomics Grand Challenges webinar series. This is our first in 2016, and I see that my slide is already in error, still showing last year. But today we're going to talk about large-scale metabolomics flux analysis. With me online are our presenter for today, Dan Jones, as well as our host, Dan Bearden, and myself. Many of you may or may not know that I recently moved to the University of Missouri. I'm now a professor in the biochemistry department here, and this is my first official webinar from the University of Missouri at Columbia. So I'm happy to be here and happy that you're here with us today. Okay, trying to advance my slides. There we go. So as you may know, this is a series of webinars that we've put together. It was originally initiated as part of a metabolomics net research coordination network grant provided by the National Science Foundation, and we've already had a series of presentations here. And today, January 6th, again, we're going to talk about large-scale metabolic flux analysis, and then we have 3 more in this first series that we would like to complete. We are still looking for volunteers who would be willing to host or present and lead the discussions on the exposome or environmental aspects of metabolomics or grand challenges that they face, as well as data interpretation. So if you are willing to be involved with that, please send me an email, sumnerlw@missouri.edu. So just a few logistical questions or comments before we get started. We ask that you sign in using your full name. This provides us a record of who's online. If you've signed in under an alias as 123, this is not real helpful for us, but we'd ask that you log off and re-log on using your name so that we can have a better idea of who's online. So there are multiple ways to converse with us or to interact. One is through the chat window here. So if you have technical questions, if you'll enter chat comments there, we will try to help you, Dan or I, as we go through this. If you have scientific questions, there's another alternative that we ask, and that is that you use the question mark or raise your hand up here during the discussion sections, and we will try to work around and get to you and get your questions and comments. So during the first 15-20 minutes, everybody will be muted as we work through the presentations, and then at the end, after the presentation, we will open this up for discussion. So again, we'll have a short presentation, and then really we are trying to build and understand a public opinion. So we would appreciate and we encourage your feedback. Typically, we've planned these for an hour. Many of them have gone over to that at 1.5 hours, but as we get to that 1.5 hours, I will start trying to wrap things up. But again, our goals are to discuss, vet, and prioritize the grand challenges, and that predominantly will be focused on flux today. And then, we want to hear you. We want to know what you're thinking. We want to take that information so that we can understand if there is a consensus there. We encourage you to interact with others on this topic, and ultimately this is meant to stimulate this interaction and really move us towards solutions to the grand challenges. So I'd like to introduce our speaker today. This is Dan Jones. He's currently a professor at biochemistry and molecular biology at Michigan State University. I've known Dan for 20 years or more, but we won't go into that. But he's a great scientist. I respect his work. He's a good mass spectroscopist as well as a good biochemist. And so this is Dan's questions or contact information if you have any direct questions that you would like to send his way or any follow-up questions. Dan has assured me that it's okay to include this information, and I would encourage you to interact with him if you do have questions. So, as part of building the consensus, we have typically included some poll questions to kind of give us a better understanding of what people are doing and at what level they're participating. And so at this point, I'm going to hand over to Dan Bearden, who will actually work us through, it's just 3 or 4, maybe 5 short poll questions. And we ask that you participate in the poll, and we ask that you participate quickly. So with that, Dan Bearden, please take over.
All right, thank you, Lloyd. So the first question is this, and you should see a quick poll on your screen with choices right now. Are you currently doing flux analysis? And that's a simple yes or no. So if you would select your answer, we currently have almost 80 people online, so that's a pretty good turnout. And over 80% of you have responded, so that's great.
So it looks like 40% are doing flux analysis where the majority is not.
Right. So good. It'll be a great opportunity then.
Yeah, so we have a split audience.
All right. Next question is, do you plan in the next year to do flux analysis? So is this on your horizon to get into in the next 12 months? All right, over 80% have responded, and it's an optimistic yes. Over 80% of you are saying yes, you're going to do flux analysis in the next 12 months, so that's pretty exciting. Next question is, come on. All right, do you think the biggest challenge in flux analysis is cost, data acquisition, data processing, access to instrumentation, or other? And at the end of the presentation, we'll discuss the poll results, and we'll have an opportunity for maybe your others will have increased by that point in time.
Yeah, but I think there's a vast majority already showing that data processing is by far the leading barrier, with over 70% there.
Yeah. All right. We'll give it a few more seconds. Okay, we'll close that one. And then the final question is, and this would be probably for those of you who are already in process or thoroughly down the road of starting it, is the availability of standards a problem or barrier to your flux analysis projects? And it's yes, no, or don't know. So if that's a question that maybe you can get that answered during the seminar today. All right. That's kind of a mixed response there, I'd say. 80% of you have responded. So we'll close this final poll question in 3, 2, 1. Thank you very much.
All right. With that, we will jump over to Dan and hand over to him. I'm going to make him the presenter, and Dan, you can get started. Okay. All right, there we go.
Okay, are you ready to go here?
You're ready. So.
All right. Well, thanks Lloyd and Dan for helping get this all set up today. And what I'm hoping to accomplish is probably aiming more at the people who are not experts at flux analysis at this stage, but I hope that this will encourage some discussion that we can have after my presentation. So to put this in perspective, my laboratory is relatively newcomers to doing flux analysis, and we've decided to take the insane approach to try to focus more on specialized metabolism in plants, although we have worked with a variety of colleagues over the years to do central metabolic flux analyses. And one perspective that I would put on this is that I see many graduate students, postdocs, and sometimes manuscripts where people have perturbed a system either environmentally or genetically, and they see a change in the level of a particular metabolite when they're doing a metabolomic study. And quite often the assumption is that if you see more, it must be that more of this is being made. And we often lose track of the idea that the levels of metabolites really reflect the difference between the rates of synthesis and the rates of degradation, and that simply the levels of metabolites often are not so easily interpreted. So the constituents of the metabolome are, almost all of them are being continually synthesized and degraded, and in many cases the fluxes, which are the flow of molecules from one particular substance to another, is often much more informative than just the metabolite levels if you're trying to define a metabolic phenotype or a change that's a result of either environment or genetic change. So, we often assume that the accumulation of intermediates of metabolites is relatively slow or is minimal compared to the fluxes through these intermediates, and for very efficient enzymes, especially those that are involved in central metabolism, that's often a good assumption to make because these are processes, metabolic processes, that are often tightly regulated, that if you suddenly see massive changes in central metabolite levels, it often reflects a truly massive change in the overall organism's health. But for specialized metabolism, such as hormones, and in my case we do a lot of work with plant natural products, fungal natural products, and in some cases microbial natural products, there's often been just an assumption that these are end products, that these are where carbon atoms go to die or accumulate, more or less. But as we begin to delve into specialized metabolism, we often are finding that it's much more dynamic than is widely appreciated. And so so I think what we really are hoping is that the fluxes will reflect the network functions, and if we measure flux, we're getting a more clear picture of the rates of reactions that are taking place in any organism, or even in secreted materials. So the rationale behind metabolic flux analysis is often that when we have tried to engineer more complex organisms, especially, it's frequently the case that perturbing or mutating a single gene doesn't give us the desired outcomes. In the case of plants, that we don't see the massive increases in crop yields, and especially this is true if we are focused more on the synthetic part of the equation and not so much on the rates of degradation. It's often the case that if you want to increase the yield of a particular metabolite, one of the best ways to do it is to knock out the pathway that breaks it down. But again, the levels of an individual metabolite don't tell us much about rates of degradation. It's something that's often grossly underappreciated. So a common approach to investigate metabolic flux is to follow the carbon. So you can either introduce a labeled glucose, which is a nice, relatively non-toxic precursor to central metabolites, and through central metabolites often into specialized metabolism. And one way to approach this is to use just a singly labeled glucose, as is demonstrated in this this figure here. So if the first carbon in glucose is labeled, the metabolic pathways that convert this often take this first to glucose 6-phosphate. There's a rate that's associated with that reaction. A second transformation, which is reversible, can take this to fructose 6-phosphate. And of course, this red ball here is representing that carbon-13, or the heavy isotope. We take this on through fructose 1,6-diphosphate, and at this point, this gets split into two separate molecules, and the carbon is, for any single molecule, is going to end up in one of these products. In this case, this is the dihydroxyacetone phosphate at the top. And but there's also a second rate that will turn this into another 3-carbon metabolite here, which in the diagram does not contain that label. But these two are interconvertible, and then you can go on and measure the amount of carbon that ends up in these different products, and those measurements, when coupled with the measurements of the levels of these compounds, can be used to establish what the rates of these various reactions are. And then once you have the rates, then quite often the data analysis approach will involve building a model of a metabolic system, meaning basically a series of different rate processes, and then trying to fit the experimental data to determine or solve for what those rate constants or what those rates of reactions really must be. Okay, so the experimental approach that we use to metabolic flux analysis is that if we have metabolic networks, we can model them, we can use the experimental data. There are a variety of different approaches that we really don't have time to get into today, relying on either a steady state assumption, in which case the experiment is designed such that there's a constant set of metabolic fluxes, that's not changing with time, or it's done over a short enough time that there's not you're not taking into account that your organism is growing or going through different stages of development, for example. There are also a variety of approaches that have begun to become more popular of late, where they're more dynamic or instationary state, where there is a change in the level of, say, expression of a metabolic enzyme, or in the factors that regulate that enzyme. Those are more complex computationally in many cases, and much more prone to uncertainties in the rates that one gets and the fluxes that one calculates. But almost all successful applications of metabolic flux analysis have required that we know what the metabolic pathways are and what the metabolic networks are. So the better we understand the pathways, as is the case for the most part in central metabolism, the more successful we're typically going to be. Now, that's not to exclude the possibility that if your model doesn't fit the data, that you won't discover some new pathways that are useful, and I know a variety of groups that continue to do that kind of work. So this is sort of an illustration of how complex the data may get in a hurry. So if you were turning carbon into pyruvate, essentially glucose being metabolized and turned into pyruvate, which is the structure given on the left here, this is a molecule that has 3 carbons in it. And if you're labeling it, or generating it from a carbon-13 labeled glucose pool, of course some of the pool may also be unlabeled, there are 8 possible outcomes that you can get. And then the first column here shows that one outcome is that none of these could be labeled with carbon-13. The next 3 columns show that either carbon number 1, carbon number 2, or carbon number 3 could be monitored or could be labeled with carbon-13. If you're doing mass spectrometry and looking just at the molecular ion of these, all 3 of these forms would look identical because they all have the same mass. They all have the same number of carbon-13s. Then the next 3 columns, columns 5, 6, and 7, show that there are 3 different outcomes that one can have with 2 of the 3 carbons being labeled, and the final column shows what, that all three, carbons would be carbon 13. So there are 8 different possible outcomes that one can investigate here. So these are what we would call positional isotopomers. So these are the isotope isomers is in essence what we're looking at, that they're the same molecule but with different isotope labeling in different positions. And of course, if you're doing NMR analysis, NMR, because these different carbons will have different chemical shifts, you can often determine the amounts of label in each of these. If you're doing NMR, if you're doing mass spectrometry, then all 3 of these that show up as just labeled with one carbon, one heavy carbon, would look the same, at least in the molecular ion species. If you wanted to tease out which position is labeled, you may in some cases be able to do that by looking at fragment ions in the mass spectrum. And the same thing for the doubly labeled. But it quickly becomes much more complex, and this is just only 3 carbons. As the number of carbons grows, then the number of options and different compositions of your labeled metabolite also grows as well. So there have been a variety of ways to approach and quantify the labels, and again, for central metabolism, we often have lots of standards available, not always labeled standards, but one key approach to quantifying a variety of metabolites, especially central metabolites, has been to use nuclear magnetic resonance, or NMR spectroscopy. And NMR spectra can be reasonably complex, but there are a variety of multidimensional NMR experiments that help tease out this kind of information. So what you see here, there's a kind of NMR experiment called TOCSY, which is essentially a total correlation spectroscopy, and this can be done with multiple isotopes. You can look at hydrogen and carbon in particular, and in this case, these are some figures from a paper that Andrew Lane and Teresa Fan published back in 2007 in the Society Journal that showed individuals, in this case some amino acids, aspartate and glutamate, for example, and the boxes that you see that they've drawn in here represent the splitting that one gets. That's essentially coupling to carbon 13. So each carbon 13 can couple to the proton, and so the splitting pattern that you get can tell you something about the substitution patterns and the enrichments of carbon 13 in the various positions of the molecule. So NMR, especially for more abundant metabolites, is an extremely powerful technique, but it goes beyond the NMR that some of us were taught in undergraduate organic chemistry, which was usually focused more on just protons and carbon spectra. That these 2-dimensional spectra get a lot of information, and because NMR signals are really very much dependent on exactly the concentration of the nucleus, or the spin system, you don't really need authentic standard to quantify, to generate calibration curves to quantify metabolites by NMR. It's a very robust and very reliable quantitative method in many regards, if it's done right. There are a variety of limitations to measuring labeling and measuring metabolites. For organic acids and central metabolites, we often use some sort of chemical derivatization, because these are polar molecules that can't just be injected into a GC. So one approach to this is to use a derivatization called the tert butyldimethylsilylation, or making TB-DMS derivatives for organic acids. This is a good approach because in essence, it takes the polar groups, the OH groups and NH groups in amino acids, and turns them into a less polar form that can go through a gas chromatograph. And it's the same approach that we would typically use for GCMS-based metabolomics. For measuring fatty acids often don't show us in their mass spectra fragment ions that allow us to deduce what parts, which carbon atoms are labeled. One approach that's been used has been to turn these into butylamide derivatives. That chemistry can sometimes be a little tricky, but it does give more information than one gets quite often, especially for certain fatty acids from just methyl esters, for example, or silyl esters. But both of these cases, this is where we've employed derivatization to direct the formation of fragment ions, that the derivative fragments in a particular way, and different derivatives will fragment and break in different carbon-carbon bonds. And in most cases, this makes it more feasible to measure the labeling in, if not individual specific positions, but in particular regions of the substructure. So if you wanted to know, is the label mostly in a fatty acid near the carboxylate, say the first carbon, that kind of fragmentation can be driven by the choice of derivatives. But I've occasionally had students and postdocs who approach me and ask, well, can't we just buy every metabolite we'd like to measure, including specifically labeled compounds at specific positions, and generally that's just not available. You can either get things that are completely labeled, or in a few cases labeled at a few specific positions, but the more complex your metabolite, the more difficult, the less available standards are that have labels at all positions. Now, as I say, with NMR you don't necessarily need that, and if you're measuring the individual compounds by GCMS, that's probably not so necessary. But I have, we've had a variety of people approach us and ask, well, could we quantify as precisely and as accurately as possible an amino acid with a carbon in a particular location? And in some cases, that's just not a feasible thing to do. So there are a few caveats that I've drawn from a paper from a few years ago from Nicola Zamboni, who basically commented that flux estimates can only be trusted if the model sufficiently describes the metabolic network and all the underlying assumptions are met. And so again, if you know what you have a good idea of what looks like, especially for central metabolism, this is usually a good approach. For those of us who are trying to extend this to specialized metabolism, often we don't know what the pathways are, we don't know what the intermediates are, or the genes or enzymes that catalyze these reactions. But where there's still some value in doing these too. And then for most flux measurements that are done in culture, say cell culture or microbial culture, it's usually the most common approach has been to use a steady state assumption and to assume that both the metabolic state and the isotopic states are steady states, so that all the intermediates and concentrations and fluxes are constant throughout the duration of the experiment. Okay. Some of the things that we and a variety of other laboratories are interested in is focusing on the labeling of larger specialized metabolites. And this can be a real challenge depending on what your labeled precursors are doing. We often use labeled amino acids and in some cases labeled carbon dioxide, grow plants in these conditions and then follow where does the label go, how much label gets into the total molecule and how much gets into the different fragments. There are lots of different isotopologs. In this case, the isotopologs are the mass isotopomers are the different masses. So in this case, if we looked at the in this particular acyl sugar metabolite, mass to charge 681 would be the monoisotopic, or essentially the unlabeled form. And then all of these others are formed after growing a plant under carbon-13 CO2 for a very short period of time. So we see as these abundances of the labeled forms grow in that the carbon is being incorporated. And one thing that we wanted to do was to assess, well, where is this being incorporated? Well, this has, 29 carbons in it, so there are lots of possibilities. If we were to take all of the different peaks and do tandem mass spectrometry, we'd be spending a lot of time and not necessarily learning a whole lot. So one approach that we've developed and essentially have followed the sort of data-independent approach is to take all of the ions that you get in a mass spectrum and induce them to fragment through collisional-induced dissociation, and to do this at different energies. So at low energy, you can knock off certain groups. As you increase the collision energy, you get quite often to smaller and smaller fragments as I go counterclockwise around this central structure here. And so by using energy resolution, even though you often have lots of other things in a crude extract that overlap, you can use the collision energy to resolve these out and distinguish, overlapping compounds and measure the labeling in each one of these. And what you see here is that, in this case we're growing these under CO2, so we expected everything would be labeled roughly equivalently, and for the most part that was generally true. Now, traditionally there has not been a lot of software that was available and designed for analyzing such complex data sets, but there is some new software, open source software called X13CMS, which is essentially a variation on the very popular XCMS software that employs an untargeted approach that allows individuals researchers to make comparisons of differential labeling in a variety of different metabolites. Okay, so in specialized metabolism, as I mentioned, most of the pathways are probably unknown. We also don't know much about what the intermediates are. So in some cases we use an initial stable isotope labeling experiment to try to establish, well, which of these metabolites are even derived from a common precursor. So one approach might be to feed plants or cells or cultures a single amino acid that's labeled with carbon-13, and then to qualitatively identify which intermediates are labeled, and to demonstrate that if the experiment and interpretation is done right, that that amino acid is a precursor of certain parts of this molecule. And once we have this information, then we can use this to start following fluxes from one metabolite in the pathway to the next. But there's a lot to be done in this area, and I think it represents one of the really grand challenges to move metabolic flux into areas where it hasn't gone before, largely because of our limited knowledge about pathways. If we want to extend the scope of measuring stable isotope incorporation, there are soft ionization methods such as electrospray ionization that allow us to measure incorporation into larger metabolites. This could be done for triglycerides and very large complex lipids, for example, providing that the analysis is designed to distinguish one lipid from another. Again, the complexity of the spectra can be quite high, and having good high mass resolution is extremely useful. There are others who, one of the aspects of flux is that we often have to ask, well, where does the carbon go if it's not going into metabolites, or how fast do metabolites get turned into components of proteins? So one approach to this has been to do an entire protein hydrolysis, such as with hydrochloric acid, and then analyze the labeling patterns in the individual amino acids that result. One area that I think is ripe for information, for useful information, although perhaps bounded by software capacity, is to combine measurements of stable isotopes with metabolite localization, such as mass spectrometry imaging. This is something we're aiming to do as our data analysis tools advance, but I think this is within reach, if not now, at least in the very near future. There are some substantial data processing barriers. Many laboratories develop their own data processing software in-house. Some of this is available shared, and in some cases it may need a software platform such as MATLAB to analyze these. And there are a growing number of open source software resources that are available, especially for processing mass spectrometry data. I think for 2D NMR, individual laboratories often have developed their own resources, and I'm less aware of tools that really are ready to go for the novice user for 2D NMR. But most novice users probably need to ought to be developing an understanding of these 2D NMR analyses before they put too much trust into a data processing algorithm. And here I've shown just a few open source software tools for metabolic flux analysis and fluxomics. There are numerous recent papers that have used these, and anyone who's interested, I mean, I hope that we can perhaps stimulate some discussion about the state of the art of data processing. So to wrap things up, what are my perspective on where the grand challenges in large-scale metabolic flux analyses lie is largely in standards. So in large scale can mean a couple of things. One could be just increasing the number of samples to do large numbers of samples, and then to do flux analyses, probably focusing on central metabolites. I think that those are things that the tools are probably reasonably good at this stage, certainly better than when I started doing labeling analyses many years ago. For more complex and especially larger molecules, the number of labeled substrates and even unlabeled standards for specialized metabolism are often just not available, and in many cases we're left to purify our own standards, to try to, for number one, quantify them and characterize them, because, as I say, in many cases the identities of the compounds are unknown. When you're doing labeling experiments, the complexity of both mass spec and multidimensional NMR is huge compared to what you're accustomed to if you're doing unlabeled experiments now. And I mean, there is some capacity to do mass spectrometry for measuring isotope enrichments at specific positions in a molecule, but where possible, this is where NMR is really strong, and for those of us who have access to both mass spectrometry and NMR resources, I mean, we have some advantages compared to those who have only one tool. There are data processing software, as I mentioned, and tools that are emerging, and we hope that continues to advance for extending things to large scale, which really means more pathways. That often means that we need more information about what the pathways are in metabolic transformations. I mean, we often, for example, know that there are a variety of certain kinds of metabolic transformations that are involved in making metabolites, but there's lots that we don't know yet. As far as data standards and reporting, this is, of great complexity, and it's something that I think we have perhaps as a society and as a community not focused on as much as we might, but I think for those who are active in this area, I hope that we can discuss this some more. There's lots of room to integrate both mass spec and NMR data. This has been done in individual laboratories, I think, that are comfortable with both, but I think there's a lot we could do to make this simpler and not require experts down the road. And likewise, to integrate flux analysis with transcriptomics such as RNA-seq is also an area that I think we can improve upon. So I think I will wrap things up at this stage, and we'll open the rest of the presentation for discussion.
All right, thank you very much, Dan. Great start to the conversation. We do have questions online, and we do want to review the poll outcomes, and then I have several questions myself. So, Dan, how would you like to proceed?
I
thought, yeah, go ahead, Dan.
I'm sorry, there's two Dans online.
Yes.
I'll defer to the speaker.
No, I was going to defer to you, Dan, and I thought perhaps we should go to the poll and then go to questions.
Okay.
Sure, that'll be great. All right. So here's the results from the first question. In this community that was online when we first started, it looked like it's almost split, but about 40% of us are currently doing flux analysis. So that was a big number. I thought that surprised me.
Yes.
So.
Well, historically, we've been pulling specialized audiences when people are working in the area of the grand challenges. We seem to have a higher representation of that group. Overall, in the metabolomics community, I wouldn't believe that 40% or 40% plus are doing flux types of analysis. So I think we have, again, have an enriched group today.
All right.
Okay.
Do you plan to start flux analysis in the next year? Over 80% of the people responded yes, they have current plans to get into this, so I think that's encouraging.
Yeah, so again, a very enriched group here. So.
Yeah. Okay. Okay, the biggest challenge. Most people landed on data processing,
Yeah.
which isn't too surprising, I suppose.
Well, I would think it's that and availability of standards. Positionally labeled standards is pretty important. So.
All right. And then the last question was, here is availability of standards a problem or barrier? I presume maybe the don't knows are people who are not yet involved. So.
Yeah. So.
Huh.
Yeah. So again, I think there is a very strong opinion from our speaker as well as our audience that standards are a critical issue. So. All right. That was the last of the poll questions, correct?
Correct.
All right. So maybe if Dan Jones, if you would put up your last slide again, I think that's a good place to kind of stimulate discussion, and we'll go to the online questions. If you haven't asked a question yet, we have plenty of time, so I'd ask you to type in your question, and we will work through those and let Dan, will you do the questions?
Certainly.
Oh, well, I meant Dan, again,
Okay.
a problem. Dan Bearden, he will read them and then he'll have to unmute them, or he can unmute them and let them ask the actual question.
Okay. Let's start with Biswap Arias has several questions. So first one is, given each model organism would have a differential flux rates, branching points, metabolic dead ends, feedback modulations, and so on, how does metabolic flux analysis, how's it going to be addressed when each organism might respond so differently?
Well, I think that's a very good question. I mean, I think the questioner certainly has hit one of the issues, right on the head with this question. I don't know that in many cases that, and I'm going to respond from the perspective of the plant work that we do, is that almost every organism we look at has quite different fluxes in both central metabolism as well as in specialized metabolism. And I don't know that, we haven't gone to the stage where we're building sort of the kinds of models yet that we can explain why these are different, but what we have done in genetic mapping is quite often is to recognize and identify mutant versions of metabolic enzymes that either have lost a regulatory domain or have, simply are not as efficient catalysts as they are in our more traditional model plants. So I certainly believe that as we go from model organisms to non-model organisms, which the genomics revolution is making much more possible, we are going to probably conclude that we can't draw too many generalizations until we understand more about what controls these fluxes. So I guess my take on this is that the fluxes the flux analysis will tell us when we can generalize and when we can't.
Okay.
All right. Mr. Perry, did you want to say something, or?
He says in his comments that he's in the lab and doesn't have the ability to do voice. So.
Okay. All right. Fair enough. Let's go to a question from Jae-Young Kim. Can metabolomics flux analysis be applied to clinical samples, FFPE, frozen tissues, or urine specimens? So I think maybe, are there clinical applications?
Well, I think there are clinical applications, and I know that that in particular one of the, and there's a nice paper by the group at Kentucky. I mean, Teresa Fan, Andrew Lane, Rick Higashi, and groups that where they introduced, they gave cancer patients labeled glucose before they went in for surgery to remove a tumor, and then they did. They measured the label incorporation into different pathway metabolites to try to understand how the tumor generates energy. And so, I mean, there certainly are opportunities to do this in terms of disease. In terms of taking a blood sample or a urine sample that has not been from an individual that has not been given a tracer of some kind, I mean, I think that it's too late to do it at that point. But I know that, I mean, going back, I mean, almost 30 years ago, there were some things that I did with Andy Clifford, where we were trying to assess vitamin deficiencies, and we were giving labeled vitamin A to individuals, drawing a blood sample the next day, and then measuring the ratio of the labeled to unlabeled vitamin A. And this isn't necessarily a flux measurement, but in its initial design, it was more a way to try to assess body stores of vitamin A. But what we found in one particular individual was that after giving him the labeled vitamin A, we went back to and drew a blood sample the next day, and the ratio of unlabeled to labeled vitamin A was just astronomically high. And of course, there were two ways to interpret this. One was to interpret this as meaning that, oh, he must have a liver that's, almost all vitamin A, or the second interpretation, which turned out to be the right one, was that he was incapable of absorbing vitamin A and getting it into the bloodstream. So I think there are lots of ways that we can use tracer compounds. I mean, my suspicion, though, is that we still have a lot to learn about clinical samples that we can do just from unlabeled metabolomics. And when we reach the point where that's not giving us answers, then we probably will want to do some sort of labeling, flux measurements to assess, say, energy, flow of energy through, say, mitochondria or other pathways of biochemical energy generation.
All right.
Jae-Young, did you want to add anything? Your mic is on.
No, so can you hear me?
Yes.
Sorry.
Yes.
Yeah, so I'm doing the clinical research, and I am thinking the metabolite and the metabolomics flux signature as to monitor the drug treatment and response of the patient. So we have frozen tissues from patients like cancer bladder and prostate cancer patients. So I'm wondering whether we can use it for the clinical monitoring for the drug treatment for the patient. So I talked with many metabolomics experts, but I could not get a very clear idea how I can initiate the work. So the many people just say, but they can do it, but it's not really clear to me. So do you have any recommendation how I can initiate the experiment or the project, since we have the lots of patient samples of frozen tissues and urine and blood specimens? Any recommendations?
I mean, my thought is that the frozen tissues that you already have are probably too late to use metabolic flux, although you might be able to do some enzyme assays, but I think metabolic flux might be overkill there. But if you have patients that are being treated, I mean, it would not be unreasonable to give a patient something like a dose of carbon-13 labeled glucose, and then to measure how well that gets converted. Now, again, as I mentioned earlier in the presentation, doing metabolic flux analysis is most easily interpreted when you have a well-defined metabolic pathway or network that you're focused on. So if you believe that the drug is doing something to impair central metabolism, then I think a flux analysis might be appropriate. But I would probably begin by asking, are there differences in the central metabolite levels before embarking on that? Because the answer may be there in the samples already.
Right. Okay, got it. Yeah, maybe if we try the prospective trial, maybe we can try it, right?
Yes.
Got it.
Okay, thanks. Let's go to Roland Wogmuth. You mentioned the limited availability of labeled and non-labeled standards as a grand challenge. What actions do you suggest to work on that challenge, for example, by the Society's Metabolite Standards Initiative? Let me see if I can unmute Roland here. There he is. Hey, Roland, are you there?
Yes, can you hear me?
Yes.
Perfect. Yeah, thanks for giving me the opportunity to ask these questions. I'm working at Aldrich work, and I have started actually about 2008 an initiative to exactly work on that problem, but I see a big discrepancy, let's say, maybe between the global needs and let's say our work, and of course the MSI. There are many initiatives globally. My question is, what actions do you suggest, do you see to work on a global level?
Well, that's also a very good question. It's just, I mean, in general, I mean, the availability of standards, whether they're labeled or unlabeled, I mean, in my opinion, continue to be a serious problem that we face, and it's historically been difficult to persuade funding agencies around the world that this is worthwhile. There are some, I mean, when the demand has grown, so I mean, it labeled glucose and sucrose and central metabolites are very available now. I mean, compared to where they used to be, and I've known of numerous groups that have embarked on essentially growing microbial cultures to synthesize specific metabolites from labeled precursors. So I don't know that central metabolites, other than those that are labeled in specific positions, is necessarily the problem. What does concern me, at least, is that if there's not sufficient demand, the producers of these may discontinue. And I've seen, over my career, many things that were available as standards no longer be available unless you know who managed to have the foresight to round them up and purchase them in advance. So right. My hope is that both the part of what the role of the Metabolomic Society and other scientific organizations will be is to encourage cooperation between laboratories that have these, because I mean, I know my laboratory now, we probably spend almost a third of our effort into purifying novel compounds and archiving them and using them as enzyme substrates to further down the road. But then once we're finished, these are archived, and yeah, I would just encourage that we should use the resources of the society to continue discussing this among one another and to facilitate communications between researchers around the globe.
Absolutely. Yeah. No, I think that would be a great thing. I don't know whether the Metabolite Standards Initiative has some kind of platform just to, let's say, to enable to make known something is a grand challenge.
Mm-hmm. So as far as the Metabolomic Standards Initiative, I think there is some area work there. I don't think it's really focused much on labeled, stable labeled compounds as opposed to just regular unlabeled compounds. But there is discussions in the Metabolite ID group as well as in the standards groups of trying to provide more cooperative exchange of authentic standards and or spectral type of data that would allow others to expand their identification confidence in numbers. There's also another, the National Institutes of Health here in the U.S. have invested money in 6 national cores, and one of those is at Research Triangle Park at RTI at the institute there, led by Susan Sumner. No relationship, but she's running a synthesis group, and their goal is to provide national service to synthesize standard materials. Now, I don't know if she's really done any synthesis of label compounds, but she might be a good contact for those that are needing specialized labeled compounds. And but I think she might also be much like other companies, that it's going to be an issue of demand and supply. Can she really afford to invest in, a chemical that's going to take 2 months to prepare and, only serve 1 or 2 users? But I think these are all good questions and comments and starting points for the discussion.
Mm-hmm. Yeah, no, that's excellent. Thanks a lot for this suggestion. I talked also with the people with the Metabolomics Workbench. I think that's a great initiative, and we are actually at Sigma-Aldrich Merck. We have all our global research development centers are working on metabolites, and as you know, it's a lot of work to develop synthesis and analysis methods for each of these. I'm just thinking now on a, let's say, on a global scale, it might be a great opportunity for the Metabolomics Society to really be the platform on a global scale.
Yeah. Well, I think that's our goal of all of us, and it's certainly the goal of the NSF that's funding this research coordination network, is to get us talking, and through this that we have a communication pipeline, and that will lead to greater efficiencies and exchange.
Okay, great. Right. Thanks.
All right, let's go to the next question from Lang Li. Would you please explain a bit more about integrating flux analysis and RNA-seq? Let me unmute Lang Li. Are you there, Lang Li?
Yeah.
Did I get your question right? You need more information about integrating flux analysis and RNA-seq experiments?
I mean, Lian, from my perspective, and this is a lot of what we're doing in my group at the moment, is the design of an experiment to do both flux analysis and RNA-seq is probably the most critical, because in many cases the transcript changes are from, say, an environmental change, it could be a drug perturbation, fall ahead of the metabolic flux change. So finding the right experiment design to time when do you collect your RNA and when do you do the flux measurements. In a microbial culture, I think it's fairly straightforward, but you're often doing this in a sort of a steady state culture if you're doing a flux measurement. And so you're hoping that you're not perturbing the RNA, the transcript levels. But in what we've generally seen is that you'd like to be able to collect RNA at multiple time points and then sort out and then try to correlate is the level of expression of let's say a metabolic enzyme gene commensurate with the rate that you measure in a flux measurement? And I think the answer is, well, sometimes it matches up well and sometimes it doesn't. And we don't, most of our experiments, we haven't done enough time points to measure the transcript to really judge where things are. I mean, I'll give an example of this where we were looking at plants making certain metabolites, and what we found was that the rates of synthesis that we measured by following carbon-13 tracers were we really showed almost no correlation with the expression of the metabolic enzymes that we thought were important. And what that eventually led us to was a recognition that there was a futile metabolic cycle, that an earlier pathway intermediate was being made and then degraded, and so that ultimately the flux through the entire pathway was mainly constrained by a gene that we didn't know existed. And so when we have well-defined pathways, then I think what we're really asking then is are there other regulatory factors? Is there some feedback inhibition, for example, of an enzyme that we wouldn't capture if we were just doing transcript analysis?
Yeah,
Does that answer your question?
Thanks, Dan. Yeah, this is Liang Li. I just wonder if doing the enzyme level measurement is more directly relevant to the flux measurement than the RNA-seq experiment. Because,
I guess.
Yeah.
I mean, I think the flux measurement experiment ideally is trying to tell you what's going through the enzyme. Now, if we extract the enzyme from an in vivo environment, then the factors that regulate the enzyme may not be the same if we've extracted it and do an in vitro analysis. And so I think that the flux measurement really gives us the most direct measure of what the enzyme is doing in vivo. It doesn't really tell us how much enzyme is there. It doesn't tell us what the enzyme would do if it were in a different environment. And so at least my own preference is I'd like to know what the enzyme does in vivo, and flux is one of several approaches that one can use to get that.
Yeah, the in vivo measurement, I guess you can use potentially quantitative proteomics approach to measure the enzymes. Yeah, that's the point I want to make. That I actually did some work of correlating the metabolite levels with the enzyme levels. There's some interesting correlation. Of course, like you point out, that the metabolites are actually being formed by other pathways, were also being degraded at the same time to other metabolites. So you need to measure the upstream enzyme and the downstream enzyme, and then try to figure out what's going on there in terms of the correlation between the metabolite level versus the enzyme levels. But with the flux measurement, I think it's not just the enzyme level right now, the activity of the enzymes.
Right, you're really getting a more direct measure of the activity and not so much the level.
Yeah, that's right. Yeah, thank you.
If I could add one extra comment. All right. Let's see. I'm trying to find someone.
Can I add one extra comment to that last discussion?
Sure.
All right. Dan Jones, you mentioned many times during your presentation that you had to know the pathway, but in that discussion you provided a good example whereby measuring the flux led to the discovery of an unknown component of that pathway. And I think there are several examples in the literature of similar type things. So my comment was that we don't always have to know the pathway, and often there are unique discovery opportunities in those where we think to know the pathway, but the modeling just doesn't meet or doesn't equal the experimental data. And in those cases, it gives us directions or insight that there may be a problem with our understanding of that pathway.
Yes, and I think that's where some of the great scientific opportunities lie, is in using these kinds of tracer analyses to uncover that perhaps our understanding is not as complete as we'd like it to be.
Yeah. All right.
There's a couple of hands up, so let's. I'm going to unmute Dan Raftery. So Dan, did you have something to contribute along these lines?
I don't know if you can hear me.
There's 3 Dans online now.
Yeah.
So.
Dan, Hannah, Dan. Yes, I think I heard you, Dan Rafferty.
Yeah, I just wanted to know if the slides are available. Sorry, I'm coming in very late, so I thought it was.
So
I would have no problems with making them available, and I'll let Lloyd and Dan sort out how that's best accomplished.
So we're recording the webinar as well, and that will go up on a YouTube channel and a couple of our websites as well. So historically, we've added PDFs at those different locations as well.
Great, thanks.
Is that it? All right. Jeffrey Moray has also got his hand up. Let's. Jeff, did you have something to say?
I just making a comment that you could help expand the limits of mass spec by maybe doing MS&E and ion mobility. Have you considered that? Because I noticed you in your labeled sugar there, you talked about doing energy resolved fragmentation, non-selective CID. Well, that's MSD to me, and then you could incorporate ion mobility after that, and maybe help you to identify these different isotopes and fragments a little bit better. It was just making a comment.
I think that's a very good idea, and that's something we're hoping to do over the next couple of years here. If there's one limitation that I've seen, at least with the current state of ion mobility instruments, although they continue to improve, is that the ion mobility resolution is, from my perspective, about a factor of 2 from where I'd like it to be. We've had some success distinguishing isomers with ion mobility, but in many other cases, the differences are too small. So if about a factor of 2 improvement in mobility resolution, I think would make a huge difference for being able to distinguish the range of metabolites where most of us work.
Yeah, something tells me that's not that far away.
I certainly hope it's not.
Yeah, that's all I have to say. Thank you.
Dan Jones, there's a request to show the slide that has the software, freely available software. If you could pull that up, that would be.
If I can move back to that. So here are a few of these.
Right. Okay. Thanks. Let's see. I don't see any other hands up at this point. So let's go to another written question. Tagreed says, how to overcome the low concentration of exometabolites released into the environment. Let me unmute him and there. Tagreed, can you expand on your question? Sorry, go ahead. Okay, all here is my cell. Okay. I'm going to mute that back. So
okay.
I guess there's the question, Dan Jones, is how to overcome the low concentration of exometabolites released into the environment. I guess.
Well, I'm assuming that the exometabolites are, all of our analytical tools have some fundamental limits in terms of how low a level they can detect. Although, mass spectrometers in the last few years have improved rather dramatically, and we routinely, I guess a couple of questions. Comment. Number one is that if you know what you're looking for, then you can probably detect it quite low. For extremely low level metabolites, and I know of a few groups that do this, that rather than using carbon-13 as a stable isotope, I know of some groups that use carbon-14 and then use accelerator mass spectrometry to detect these, because it's the same tool that's used for radiocarbon dating. And if you have a way of purifying or fractionating exometabolites, you can then quantify the carbon-14, which is at such low levels that you wouldn't detect it at using a scintillation counter. So for all practical purposes, you might consider that this is a non-radioactive stable isotope or semi-stable isotope of carbon. It allows you to get to ridiculously low levels of detection. I know some years ago we brought a human volunteer into a study, and we analyzed a blood sample from her to measure carbon-14, and it was about 2 times what the normal, what we call modern levels of carbon-14 ought to have been. And we asked her, well, did you ever participate in a study where you had been given a radioactive form of carbon? And she said, yes, 5 years ago. Wow. So we have used carbon-14 and labeled red blood cells to monitor the lifetimes of red blood cells, and in essence the fluxes of micronutrients in and out of red blood cells and in and out of different tissues. So there are commercial laboratories that do this now, and a variety of them. It's become a very popular thing in certain pharmaceutical companies to use this sort of low-level carbon-14 as a tracer.
Okay. Let's see. Let's go to Wimmel, who's in at RTI. He's had several responses here about. Oh, I don't see him online anymore. Oh, there he is. Okay. Wimmel, can you tell us more about accessing the synthesis services that the NIH has funded?
Yeah. Thank you. Can you hear me?
We can barely hear you.
Okay. Can you hear me now?
A little better, but still.
Hello, can you hear me now?
Yes, go ahead.
Okay. This NIH has funded for synthesizing metabolite standards through 2 cores. So one is RTI International and the other one is SRI International. And you can nominate compounds to these metabolomics standard synthesizing standards through the Metabolomics Workbench website. I think I posted it. You can get that. Yeah.
Okay.
And the process is actually you can nominate this, and then the steering committee they will actually review it, and then after that, if they approve it, they will ask for synthesizing this through these 2 cores.
So can you comment on how the nomination committee reviews? So if it's just supply and demand, it's less likely that you're going to get your sample approved or your nomination approved. But if it's on scientific merit, there might be a greater opportunity to get the standards that you need.
Yeah, they give the priority for this scientific merit and all this. Yeah.
Mm-hmm. But in one of your other comments, you said that there's only about 15 standard labeled compounds.
I think it is more than that. Actually, if you go to the this Metabolomics Workbench page, there's a link for standards, and you can see how many metabolites. Actually, there's it's about 40 records I can see right now.
How many?
Yeah. For I just talked to actually Jason Burgess. He's part of this synthesis, and he said it's about 15. But I can have a look at and then tell you how many label compounds synthesized so far.
But I expect it's still going to be a small number.
Yeah, that is true.
Yeah. All right.
Yeah, it's a challenge. Yes, yeah, that's true.
Okay. All right. Well, thank you for your comments and feedback. If you haven't looked at the questions, you can actually see this and some of the information that Wilmette is talking about or posted in the questions panel.
Right.
Okay. Back to Dan and Dan.
Yeah, so let's, Reza says he can comment on capturing and reporting the flux and tracer labeled experiments. So let's turn on his microphone. Reza, are you there?
Yes, hi.
Good morning or
What
good
do you
afternoon
think?
for you.
Good evening. Yeah. Thank you guys again for this nice challenge. I think I missed the opportunity when the standard topic came up, but my comment wasn't necessarily on sort of compound standard, but standard in terms of the section in your slides, Dan, which is about reporting it and capturing this information. So we started from a very small group, not to neglect the flux and tracer labeling community. Last year with Nicola Zamboni, Teresa Fan, and Martha Cascante, and a couple other people, just a small group on getting the requirements and getting what is generally reported, how to capture this information, how to capture the data, how to understand it. So I thought I'd use this opportunity, since it brings a core of flux people or people who are working and are in this area, that we now want to go to expand this and get a wider invitation that if anyone's interested in sort of, because there's a lot of work, there are so many different types, elements, techniques, technologies, methods, and it needs a really larger group to be able to participate and share. So that was a comment essentially, saying that if you get in touch via Data Standard Task Group, and I'll be happy to put you with this effort that we're doing to capture this flux and tracer label studies. And that's it.
I think that's a great suggestion, and I hope that this presentation will encourage more people to get involved and to provide their input.
So Reza, as an action item, could I ask you to post more information or a link to this task group where people can actually provide direct feedback?
At the moment, there isn't a specific website to this, but if you get in touch directly by email to me, then I'll add you to this sort of Google Documents. We haven't set up anything. We're a bit slow, like everything else, on actually making this available or public. It's always bottleneck of something slows things down, but we hope to get that in next sort of a wider asking for call or joining or whatever you call it soon. But even if you can't wait that long, or you're happy to help or use this opportunity essentially, then please join. Should I give my email, or you can
if you tell me your email,
groups?
I'll type it in real quick. Just what is your email?
Okay.
What is it?
And the simplest thing is reza@ebi.ac.uk.
Reza@ebi. Dot what?
AC.
AC.
Academic. .uk.
UK. Okay.
Right.
Okay. So I've added this to the comments and a response, and so please contact Reza directly if you want more information about the task group. All right.
So we can go back to another question that Ms. Wapper Misra posted. It also goes to genes. So in large-scale genetic screening approaches, we're using random and site-directed mutagenesis approaches in E. coli or yeast, where bioengineers generate potentially hundreds of strains. What would be an ideal approach for metabolic screening to select the strains of interest?
Well, I think that much of this can be done, and I know some groups that are doing this already, to do a very basic, if you're screening, you can feed to start with the labeled glucose and measure the rate at which the label gets transferred either into other central metabolic pathways, or for those who are doing synthetic biology and trying to select organisms to make interesting compounds, to follow how much of the flux of the glucose actually gets into the desired compound. So I don't think that there's any sort of new or necessary change in the approach. I mean, the standard metabolic flux analysis, I think, would be in many cases immensely valuable. As I say, we've found it to be very valuable in plants, and I think for there's labeled glucose is now, it's not so outrageously expensive that it prohibits one from doing that sort of large scale screening.
All right. Let's see. Let me go and see if anyone's typed a.
All right. Well, while you're looking, Dan, I'd like to ask another question, and really it comes down to scalability, and there's been some discussion about the effort that comes to it, but on an omics level, how feasible or how scalable is fluxomics at this point in time, and based on your opinion?
I guess a lot depends upon just what the objective is. I mean, if the objective is to build a more predictive model of how a whole organism works, we often have a long way to go. There's so many regulatory aspects that I don't think we understand as fully. But on the other hand, just to the last question, could we use flux analysis on a large scale to screen yeast or bacteria to optimize a particular process? I think that the main bottleneck right now is finding the right software tools to accelerate the analysis. It's not that the analysis is difficult, but so many groups, when they're new to this, there's a bit of a learning curve to appreciate, number one, how to design the experiment properly, but also how to do all of the analyses. Once you learn to do these kinds of analyses and have the software tools and the familiarity to use the software tools, I don't think it's terribly difficult to do. And I would hope that many more people will explore this, and that as new software tools become available, that either to embrace the people who develop this, or to find collaborators that can do this analysis if it's not something that an individual laboratory has the expertise in.
So again, data processing is one of the key challenges of large-scale flux analysis, and that kind of showed up in the poll. And
I think most laboratories, if they either have good NMR capabilities or good GCMS or LCMS capabilities, probably already, if they're doing metabolomics now, they probably have the analytical tools to generate the data sets. It's more the question of what needs to be done to recognize what the bottleneck is. But in many cases, a fairly elementary and rudimentary flux analysis model will highlight bottlenecks in metabolism without having to go through an immense amount of data information. It's just that we haven't always had the tools ready to plug and play.
Yeah. So it's still. Go ahead.
I was just going to say there's an interesting question from Will Thompson. I don't think he's still online though. So, it says, is there a place for using broad spectrum or unbiased metabolomics where not all the pathways are known, but a tracer is used as an input molecule? As an example, if he suspects something may be going on with dysregulation of methylation pathways, but we really don't know what the metabolites may be playing key roles from a standard differential metabolomics study, can we use C13 metabolomics or MET or C13 SAM and then look for anywhere a heavy ion may be dysregulated? Seems like a data analysis nightmare, but I'm wondering how the panel views this sort of pathway fishing.
I guess I think that's a very useful approach to use, and I mean, especially if you have something like labeled SAM, which is already available. I mean, for us, I mean, one of the limitations is getting labeled SAM or unlabeled SAM, for that matter, into the cells or into the tissues where the metabolism happens. For microbial cultures, it's probably less of an issue than it is with plants, but if if the precursor can be delivered and then incorporated into the metabolic network, then it's a very approach. And I don't think that for that kind of analysis that the data processing is nearly as daunting as it is to construct more sort of flux balance type of models that you're trying to explain all of say, central metabolism. I mean, if you're looking for differential methylation patterns, it's fairly straightforward, even with with a tool such as XCMS, without the C13 version, to extract out and find where differentially methylated or differential incorporation of carbon from SAM is showing up. I don't think that that's a terribly difficult approach, a difficult set of analyses to do.
Yeah. I would agree that there have been several discovery type of publications in the literature showing exactly that, and Dan's answer is much more sophisticated, but I would just concur with him. And note that we have 5 minutes, maybe time for one, maybe two more questions.
Yeah, if you would raise your hand if you wanted to contribute or make a comment, that would be very helpful.
Final comments? Yeah. All right. No more questions or comments? Dan Jones? Dan Jones, would you like to make some closing comments then?
I guess my closing comment would be that this is, I think, as our poll shows, that this is an area that has attracted a lot of interest. And I mean, my advice to those who are new to the field is that they should discuss, especially the data analysis, with experts, and recruit collaborators where they can, because that solves a lot of problems to have people who have done these kinds of analyses look over your experiment design and look over your plan to do the analysis. So I mean, that for us, I mean, we have a number of very good people here at Michigan State that I've relied on, and to get our laboratory up to speed, and I would hope that everyone else can find some equally useful collaborators.
All right. Well, thank you, Dan. Thank you, other Dan, and thank you everybody else for joining with us to talk about the grand challenges that we face in metabolomics, and specifically in large-scale flux analysis. Again, this video will be available online, and we will post Dan's presentation slides independently as well. So thank you again for everybody's time. We'll bring this webinar to a close. My co-organizers, I'll stop the recording here shortly, but I want a trailer of silence, and we'll come back on and do a wrap-up. So take care, everybody. Thank you now. Have a great year.